We don't know what the limits are. It's not size. I think the limits are solubility in the vesicles themselves. We have applied different, what we call fusion proteins that we can fuse things together with to either target them or improve their solubility. And in the case of complex polypeptides for protein folding and disulfide forming, the disulfide bonds that help hold the three-dimensional structure of a protein in place. So these tools need to be applied empirically so we understand what's going to be the best system for the best target.
Today, we're exploring a new approach to vaccine delivery, recombinant extracellular transport vesicles. Christopher Locher, who is the CEO and co-founder of Versatope Therapeutics, joins us to unpack this nanovesicle platform, how it differs from traditional vaccine delivery, and what it could mean for broader, more durable immunity. Christopher brings drug discovery experience from Vertex Pharmaceuticals, Opsona Therapeutics, and Maxigen. Let's get into it. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Christopher. It's good to have you on today.
Thank you, David. A pleasure to be here.
Let's start out a conversation with perhaps this a bit controversial question. Share something that you believe about bioprocess development that most people disagree with.
So one of the things I found out in the process, I didn't do this early on, is that one can actually make a clinical batch for phase 1 human studies early on by using a simple process, and it doesn't have to be GMP. This is especially true if one does a clinical study overseas. The FDA in the United States is getting on board with this approach where you just take a handful of subjects, but you don't need all the bells and whistles and millions of dollars that go into process for a phase 1 clinical study. That can be done concurrently or later when one raises more money for a specific program.
That's interesting. That's the first time I hear about that. Very interesting approach indeed. Before we dive further into drug development, let's start out with your story, Christopher. Tell us what was the initial spark that drew you into biotech and what were some interesting pit stops along your long career leading now to co-founding a company?
So there were several milestones. I think early on, I was always inspired by my teachers as early as high school. I had very dynamic and engaging, entertaining teachers in science that kept my interest and focus in learning about chemistry and biology. I've had waxed and waned over the years, but I think overall it was my teachers that inspired me to study biology. And I think it was also my active imagination about what it would be like to work in infectious diseases and tropical medicine over the years. So that's why I got into vaccines to begin with, because there still remains a huge unmet need for parasitic diseases, for example. Not so much in the developed world, in Europe and North America, but below the equator, I think is still problematic. There are control measures that are being used that are still pretty antiquated. I know mosquito bed nets are still the best prevention for diseases like malaria. We can apply new scientific tools to solve these problems. Research in the United States, a lot has been focused on this. There's quite a bit of an effort. When the United States had these problems of things like yellow fever and malaria, we've since eradicated them due to engineering measures largely and putting screens on windows. But my point is that there's still an unmet need all over the world. And I was inspired, I think, to start the company because I had been in industry for a while and I still saw the problems with drug delivery. We tend to have a Descartesian view of deconstructing everything down to the atomic level, which was great when I worked at Vertex Pharmaceuticals because we did structure-based drug design. And there you modify specific atomic groups, rearrange atoms, molecules, certain functional groups to improve the binding of a target. That's all well and good, but then there's another element that getting that drug to the target and making it stay on target is often done with vesicles, such as exosomes or nanovesicles in the body. So things are gobbled up and spit out, and the microbiome in the human gut also delivers vitamins and nutrients to our body. They can also deliver diseases. So when I first saw the Versatope technology, I was inspired by nature largely because these little nanovesicles can be engineered as immunomodulators to bolster the immune response or attenuate it. So inflammation and anti-inflammation are really two sides to the same coin. And by using genetic programming, we can program it to be either pro-inflammatory or anti-inflammatory. So having my background in biotechnology and working in these different disease areas, I think was part of the insight to see the opportunity, not only for vaccines, but immunomodulation overall.
Tell us more about these vesicles. What kind of technology are you using? How do you produce them? How do they work?
So we produce them in microbial bioreactors. When I talked earlier, you can also make them in shake flasks, just like shake and bake kind of approach. You harvest the supernatant or the fluid on top by removing the cells. And in the supernatant itself has little tiny vesicles. So these tiny vesicles can be engineered so that they're relatively uniform in size and consistent in batches, especially when one uses cloned and single cell approaches for expanding their products. And it's this approach that we've used early on to genetically engineer these so that the lipopolysaccharide, which is a toxin in people, can be modified so it's no longer toxic. That's hampered a lot of efforts in using these types of vesicles to really attenuate these to the point where they no longer pose an issue to inflammation. So again, it's a balance from inducing inflammation and reducing it and inducing it at the same time. So these can be fine-tuned accordingly, and we can program these to express vaccine epitopes in the case of vaccines so that they're displayed on the surface of the nanovesicles, or they can be engineered to express antibody fragments for cell targeting. And those are called paratopes. You have the antibody functional domain. That's of high interest to me because these little vesicles and the microbes themselves are orally bioavailable. Instead of getting an injection, one could potentially envision with a little bit of imagination that you can deliver antibodies by the oral route using this approach.
Oh, interesting. So theoretically, tell me if I'm wrong, could you co-produce your protein of interest, add your vesicle at the same time, and then purify and use that as a drug, or is that a bit more complicated?
No, that's exactly what we do.
Oh.
And that's what we prefer doing. You can also conjugate and package drugs in the vesicles as a two-step process. That's been done. That's doable. I like the idea of doing everything in one go and expressing the drug as well as the vesicle in one go in a single process so you have a single product. That's how we make our vaccines.
And what kind of products can you co-produce? What are the limits?
We don't know what the limits are. It's not size. I think the limits are solubility in the vesicles themselves. We have applied different, what we call fusion proteins that we can fuse things together with to either target them or improve their solubility. And in the case of complex polypeptides for protein folding and disulfide forming, the disulfide bonds that help hold the three-dimensional structure of a protein in place. So these tools need to be applied empirically so we understand what's going to be the best system for the best target. It's not something that we can predict necessarily right off the bat. We have a sense of what direction to go in. So what we generally do is make a lot of different clones, screen them for protein expression, solubility, and having the right shape. And once we have that, we can scale it up and use it as a master cell bank.
What are the benefits of your platform compared to other systems?
Cost, efficiency. Exosomes are hampered by high development costs. Oftentimes they're mammalian expression systems. The yield is poor, the half-life is poor, and the commercial scalability seems to be more abstract than it is with the vesicle technology. There's already products on the market with the vesicle technology for vaccines. So we believe it's feasible to have commercially scalable product at very high levels of manufacturing, thousands of liter scale, where one can have enough doses for entire populations of people. That's in contrast with mammalian cells, and it can be done, but it's harder, and it's certainly more expensive to do it that way. With the exosomes derived from mammalian cells, they tend to be only lipid, and they're delicate. With our products, there tends to be a lot of sugar in it, and that sugar imparts a stability you're not going to get with the typical exosome or other types of nanovesicles that are made with lipids.
How do you go about developing a manufacturing process for these vesicles? What can you copy paste from more established platforms, and what are some unique features of your new technology?
It depends on the cell line we're using. Some cell lines are more hardy than others, so we've had to adapt the bioprocess in the bioreactor itself to make it more amenable to delicate products. On the other hand, if you just do things in shake flasks, it tends to be good enough for small-scale pilot studies. We're an early-stage biotech company. We're phase 1 ready, so we have reached that clinical milestone with an allowed IND with the FDA for our vaccine drug product. But we do want to also explore other areas as well, are of high interest currently and of an unmet need in biotechnology. And I like pushing my boundaries with the technology, so I like doing things like expressing antibodies on the surface. That's something that people typically don't do, but I want to do it. And I don't want to do it as just like a single antibody. I'd like to do multi-specific and bispecific antibodies to have a single product that one can develop that hits several targets and has the stability and the product characteristics for a high therapeutic index. So good PK and PD properties.
What were some challenges you have encountered as you were developing the process and now scaling up the process?
Aside from money, I think part of it was just logistics. COVID happened, so that shut down our operations in terms of having a reliable manufacturing team that could work in an efficient manner. We had to break things up and work in shifts, so that hampered our efforts over a period of several months. It also hampered our manufacturing organizations that we worked with to develop this, unfortunately. Then we also had problems in funding. Working with the federal government in the United States has its challenges. It's great because they have good support, but the money isn't always as readily available as you would like it. So there's been a lot of stops and starts, having to wait for approvals for certain things. And now with the funding rescinded and the NIH kind of rechanging its focus out of vaccines, it's been really challenging to try to raise money for our products. So aside from funding, I think that COVID and some of the logistical challenges were supply chain issues as well during COVID. That was a consequence. We couldn't get filters for concentrating and purifying our products. That was a big bottleneck. There was a huge delay time for that. A lot of products were made in China, and a lot of shipping from China was delayed. And as you know, a lot of products sat on docks for a long time in Los Angeles.
That was a huge issue for a lot of companies, the supply chain. What are other risks you're still encountering today due to a novel format, perhaps a new way of producing this? What stands out here?
I think a lot of it is education of people. People want everything proven. The goalpost seems to get being more pushed out. A few years ago, let's say 10 or 20 years ago, people could raise money on preclinical and early stage clinical products. Now everybody wants to see a phase 2 clinical study, proof of concept. And by then you really don't need a whole lot of funding because you're already off to the races and looking for partners to develop for further commercialization and licensing of the product. But now the goalpost has been moved. There's a lot more competition and there's less money available to support programs in biotechnology.
Now, I'm curious how you navigate the regulatory side, because a lot of things have happened lately at the FDA and other agencies, novel new pathways, new ways of approaching regulatory. And at the same time, you're also proposing, shall I say, new modality. So how did you go about that?
I think the FDA was very understanding. Certainly the group that we worked with, we didn't have any problems. And this was before 2025. We got our IND allowed in January of 2025. I think it was the 19th of January. And we submitted our IND for their review just before the Christmas holidays. So they turned it around in less than a month. And they had no hold clinical questions. They understood the product. They understood what we were doing. I think working with a really good regulatory team and a committed CDMO for manufacturing was extremely helpful in us reaching that milestone without a lot of hiccups. Unfortunately, I don't know what the situation is now since that time. In the last year and a half or so, I know that a lot of people have left the FDA, and that's been challenging. I've been talking to people in Europe about collaborations. So I think there's opportunities abroad that where certain doors have closed in the United States.
And to what extent did you have pre-briefing sessions, discussions with regulatory agencies to anticipate certain things?
Well, our first product to the clinic was our universal influenza vaccine, and we submitted an interact request for early interactions with the FDA when we first launched the product and the company, sent them the preclinical data and said, do you guys want to talk about it or have an in-person meeting? But we got a letter, a written response only. They said, no, we're good. We understand what you're doing. Thanks for keeping us in the loop. We opened up an IND. We had a pre-IND, asked for feedback on the safety talks design and our overall CMC. They really didn't have any questions. They said, no, just make sure your quantification and your stability tests look good and you have a good ELISA test for quantifying that, because that's what we had proposed. That's for antibody binding and measuring your actual product itself, the specificity of the product and quantifying it. So we said, okay, and that was it. Then we submitted the IND and that was it. So we've never had a face-to-face meeting. We've never had that requirement. We just asked for information only. A written response only for our IND, and that was good enough for our purposes. Now, phase 2 will be different. I think that's where we'll have much more robust discussions with the regulatory authorities.
I imagine some analytical questions and stability questions will come up then. What is your approach there? Because I imagine you have some characterization challenges, probably much more complex than just if you produced an antibody.
Well, yes, it's definitely more complex where you're dealing with a lot of different polypeptides there, a lot of lipids and sugars that can be characterized. But we have the tools now to do that. A lot of it is with mass spec. You can quantify and measure the number of proteins and how much. That was one question they had. What is this comprised of? And we're able to provide that to them. And they said, okay, that's good. So now we know what it is. It's mostly our product, our drug product. And we characterize that with ELISA, with a specific antibody. So we're able to quantify it for stability as well as the number of molecules that are present in a specific ELISA plate. Yeah, that was good enough for our phase 1.
And what do you anticipate for later phases?
Scalability. So we're going to have to show a commercially scalable process. So we're going to have to work with a good CDMO that can do this. I envision something like extended batch manufacturing. I've talked to CMC people about this, and that's something that's becoming more and more common, not necessarily continuous manufacturing, but extended batch, where you can harvest your material as you go along during the course of a week or two and keep the bioreactor running. And in that way, you don't have to scale to 10,000 liters. You can do smaller scale, like 1,000 or less. And now they have disposable bioreactor systems as well that may be amenable to this approach. But deciding on how we're going to do this is going to take some work up front. Probably at least 6 months to a year. We will need a commercially scalable process for phase 2 and beyond.
And because of the smaller scale that's required, could you envision perhaps more local manufacturing south of the equator, for instance?
Yeah, you could, provided there's stability there. I have talked to folks in Africa about the vaccine manufacturers there on the networking. They're interested in our approach. We have a malaria vaccine program that is currently bispecific, but we want to make it a trispecific vaccine that hits all 3 stages of the parasite life cycle. And since malaria is an important disease in Africa still, they've expressed interest in working with us to transfer this process to Africa. Other folks, not necessarily competitors, but people that we've worked with, I know have transferred the technology to places south of the equator. And it seems to work. I mean, provided that all the materials are there and they have a steady supply of purification equipment and filters required so they don't run into the same roadblock we did during COVID.
What I'm hearing, it's not necessary to produce south of the equator because you could produce it in Europe or in the US and then ship it to these nations.
Yes, you could do that too. And you can do that at room temperature provided the material is lyophilized or freeze-dried.
That would also simplify it because I imagine if you produce it locally, you need qualified staff, you need a stable environment, and a lot of things have to come together in order to work.
I believe for small places like in outbreaks and pandemic settings, something we've taught about people is having decentralized manufacturing where we could actually ship a manufacturing unit on a skid or on wheels, all self-contained within a single module where you have both the upstream and the downstream processes and just wheel it on a big cart but cover it in a big piece of plastic, keep it clean and plug it in and turn it on and run the machine. It is possible to do that depending on the scale. I think it would be pretty cool to do something like that.
Christopher Locher has walked us through the science behind Versatope's vesicle platform, how it's built, what makes it different, and how it could change drug delivery. There is still plenty more ground to cover with Christopher on manufacturing, regulatory strategy, and lessons from the founder's seat. If this conversation has been valuable, please leave a review on Apple Podcasts or your favorite platform. Thank you for tuning in, and I'll see you next time. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.